инфекции мы перенесли все в онлайн, и биоинформатическая секция наша, которая сейчас началась, она вообще должна была начаться с доклада нашего keynote-спикера Алана Герберта, основателя компании In-Out Bio в Бостоне, США, который к тому же является научным консультантом международной лаборатории биоинформатики. И Алан специалист в области Z-ДНК и разработал концепцию флипонов, генетических регуляторов клетки. И наша лаборатория активно работает вместе с ним. И как раз вся секция будет посвящена части этой работы, как раз подготавливая доклад нашего основного спикера. И первое слово предоставляется, первый доклад сделает Назар Бекназаров, который называется «Методы интерпретации омиксных данных в задаче предсказания Z-ДНК». Назар, пожалуйста, вам слово.
Okay, thank you. I will do my presentation in English. I hope everyone will understand this. And if there is some question, please ask me in any language, and I will begin.
Значит, Назар, 20 минут доклад и 5 минут на вопросы. Мы будем, ну, сейчас вот уже даже, может, если чуть меньше, мы будем постараться придерживаться расписания.
Okay, okay. Theme of my presentation, methods of interpretation of omics data in attacks of Z-DNA prediction. First of all, let me talk about Z-DNA itself and the class of secondary structures, the part of DNA itself. I would like to explain the task itself, what we're going to predict. We all know the canonical form of DNA. It's called bDNA, and when DNA is accessible, in most cases, the double helix DNA is in form of bDNA, and we all know crystallic structure, and we know a lot of this structure. But sometime, by some reason, we found that DNA could be found in different structures, in different forms. This structure, like bulges, like triplex DNA, when there is 3 chain of DNA together in structures like G quadruplexes, and in structure like Z-DNA and A-DNA. And my work about predicting Z-DNA. What it is? We all know that bDNA is double helix DNA, 2 DNA chains that rotate clockwise, and if you see on this outside of DNA chain, and sometime DNA chain can increase the angle of rotation, and thus we can obtain aDNA. aDNA is shown here, and we can understand why double helix DNA can increase the angle of rotation. For example, if we want to open DNA for reading, the risk could be accessible rotation angle, and this additional rotation can be stored somewhere, for example, in aDNA. And we understand why it's present somewhere in DNA, but sometimes the DNA changes the angle of rotation to -1 and turns to Z-DNA. And Z-DNA structure is much less energy efficient than B-DNA structure. And there is a lot of question why sometimes DNA could be found in these very structures. And there is some works that shows that it's not a random event. It's not some noise. It's some structural feature of some regions of the DNA, and the importance of this structure is not zero. And for example, we understand that it affects the transcription rate and so on, and that's why it's impossible to understand where it could be found and why it could be found there. And first of all, we have to say that there is not a lot of data about Z-DNA. There is some some in vivo data, 2 experiments available for human and 1 experiment available for mouse. And for mouse, in mouse case, this experiment still not published. It made for us, and it will be published in some time. And there is some in vitro experiments, but in vitro and in vivo experiments differs a lot, and that's why in vitro experiments could not be used for in vivo prediction, because as we understand now, there is a lot of additional structures that could affect DNA, and in vitro it could not be the behavior of DNA is not the same as in cell in terms of Z-DNA structure. Okay, and there is a task, and for now there is some methods that that tries to predict Z-DNA sites using only DNA sequence, and some works show that it's not enough for good Z-DNA prediction, and some additional structures in the cell could affect DNA to behave differently. And some of these structures and could be found in omics data. What omics data is? It's some additional information about DNA that could not be found in DNA itself. For example, Heisten marks. We understand that Heisten marks is involved in DNA transcription, in DNA behavior, and so on. And different Heisten mark code can modify, can affect the cell behavior and so on. And additional information like RNA polymerase, site of transcription factors, DNA sequence experiment could be a good information for the model in order to increase quality of prediction. And it's more than 1,000 different tracks, different features, and together it's more than 60,000 separate experiments, and there is a lot of data. And the main question is how we should deal with this vast volumes of data, because together in uncompressed form it exceeds 1 terabyte of data. It's for these volumes of data, it's impossible to use RAM memory, and it's difficult to use even hard disks, because for one epoch you have to you have to uncompress all the you have to deal with whole set of data, and it's more than 1 terabyte. That's why we created a some container for this, and the compression rate exceeded 10 in power of 4, and that's why we could effectively decrease the volume of data to something around 200 megabytes, and now it's possible. It was possible to have all the data in random access memory, in RAM memory. And after creating the effective container, we could talk about deep learning models that we used for Z-DNA prediction. We tried a lot of different models, and that utilize different aspects of the data. First of all, we used LSTM model, some RNN model that understand the DNA like sequence data, and can utilize information about order of every nucleotide, and can somehow find some information not in this very nucleotide but in neighbor one. Then we tried to utilize CNN model that can look for a local construction of every piece of DNA, and then we tried some hybrid model, and that uses CNN and RNN part together, and see whether it could make a better prediction. And thus the whole model, the whole idea could be shown here. We took a lot of omics data, we then sparsify it and effectively collect in memory. Then we use DNA sequence itself, somehow encode it. We used a lot of encoding techniques. Here we showed only one hard encoding, but then we added some more techniques, and then we feed all of the data to deep learning model, and deep learning model itself create two outputs. It's create a full genome annotation, de novo full genome annotation, and prediction of target sequences. And from deep learning model, we can create a model interpretation, the theme of this presentation. And here we can see that we created a good de novo prediction. Here you can see the prediction that model creates, and after creation, this prediction and showing that this prediction outperforms all current methods for Z-DNA prediction, we have to ask ourselves why the model acts and behave in this very region this way, and in for example in different regions it does not predict Z-DNA. What information this model used? And in order to answer to this question, we focused on model interpretation, and our first very approach was a simple linear regression. Let's use the simplest model that we could imagine for predicting the same stuff as the deep learning model. And we tried use this and saw that the quality of this model is lower, but not significantly low. And we understand that interpretation of linear regression model is much easier, and then we decided to focus on this. And here you can say the whole idea of this model. We just use one fully connected layer and then predicted Z-DNA itself, and one fully connected layer with no additional architecture is linear regression itself. Okay, and then we create an interpretation using only weights of this linear model. And when we used it, and here is the example of this, and then we created some additional analysis whether these results could be understood as random and so on. And we showed that it's not a random story. The most positively significant and negatively significant weights are not from normal distribution, and then we try to somehow analyze these results. And first surprise that we found was the fact that in two different experiments, we named it by some reason Z22 and flag, reveals they both reveals the one story, the Z-DNA and on human genome. And we created two different models on Z22 experiment and in flag experiment, and we saw that the interpretation results was highly correlated. And for us, it was very good sign. They have not ideal correlation. They have some problems with it. You could see there, but in general, it was a very good start. And then we created some more additional analysis to show that this interpretation is robust to noise and robust to different sampling, and this was very important. And the second step that we tried, we create the model interpretation between two different organisms. And here you can see the results. Here every point is accorded to single weights in every model. For example, for here is for human and here for mouse. And then we analyzed the correlation of these weights, and we saw the correlation, and correlation here could not be obtained randomly, and correlation with any other model is significantly lower than the correlation we obtained in these very experiments. And these additionally showed that this method could create an interpretation even between different organisms. That's why it was very interesting for us and fascinating. And then we additionally checked this method for robustness for noise and robustness for different train test divisions, and we saw that the results is robust for these changes. And then we tried to analyze the results of this interpretation and analyze the top and the bottom of this list of important features, and we saw that some of these important features could be proved by by some different analysis. For example, we analyzed whether some region have high potential of having Z-DNA inside, and in reality we saw that, for example, regions in the top of the list have some of the features in the top of the list have a very high rate of very high probability of having Z-DNA inside, showed by different methods, and some of them are really proved in different papers to have connection, to have some involvement in Z-DNA construction and Z-DNA and here is some just random example of genes and structures in cell that could be have a connection with Z-DNA, and this additionally proved our methods. And I think now I'm out of time. And this we published a paper about Z-DNA prediction, and now we're creating a following paper about Z-DNA interpretation.
Thank you, Nazar. So, dear colleagues, please ask questions. Okay, I will ask a few. So, okay. On the slide, you showed that actually there is not a high correlation between Z-DNA model, important features in model for Z-DNA prediction in human and in mice. Correlation is not that great. Why? So why we don't think that the same factors are important for Z-DNA information in humans and in mice?
Okay. We can discuss we can discuss the value of correlation in two aspects. First is the correlation itself not very high. It's something about 10%. And if we, for example, if we throw away some noise features, for example, like this, it can increase to 12%. To 12%, and it's hard to tell whether it's high or no. And then for to answer this question, could be this correlation found randomly? We tested it using using two different techniques, and we saw that it could not be found. That high correlation could not be found randomly, but still it's not 100% or it's not 50%. It's pretty low in terms of correlation when we talk about some different things. And I think that the main reason is that still these experiments is pretty different. The time difference between this experiment is more than 10 years, and there is a lot of additional stories that could lower the correlation. For example, as I understand, the experiments in for mice and for human has different tissues, has a little bit different techniques, and all of these in my point of view could decrease the value of correlation, but still in terms of randomness, in terms of p-value is high. The correlation is high.
Thank you very much. So, dear colleagues, please, any short question? Otherwise, we are running out of time. Thank you, Nazar. So,
thank you. You turned off your
Sorry. I'm glad to give a floor to Pavel Latushев with his talk about growing quadruplexes prediction. So Pavel, please start.
Yes, hello. Let me start it. Sorry. Hello, my name is Pavel Latushев. I would like to talk about domain adaptation methods in the task of quadruplex prediction. My colleague Nazar told about secondary DNA structures. I think there is no need to talk about them again. But one of possible secondary DNA structures is G quadruplex. G quadruplex is a secondary DNA structure. It forms in guanine-rich regions. Actually, it consists of 4 chains. DNA structures consist of guanine tetrads. Every tetrad is a structure of 4 guanine molecules bonded with hydrogen bond and with an ion of metal in the center. And these tetrads are stacked into G quadruplex structure. Usually there are 3 tetrads, but it can be more, 4, 5, and so on. Quadruplexes have connections with some biological processes like cell replication, translation, transcription, and other processes. So they are actively studied in biology. They connected with cancer and other diseases. We have 2 available datasets on human. The first one is G4-seq dataset. It contains about 700,000 quadruplexes. This is an in vitro dataset. It means it has quadruplexes found in laboratory condition in a test tube with stabilizing agent. So not all of them actually can be found in real cells. A huge part of it is formed only in these conditions. And the other dataset is G4-seq dataset. It is in vivo dataset. It means it has quadruplexes found on living cell chromatin using ChIP-seq method. Both datasets are not very accurate. They can have some false positive regions. So their intersection has 7,000 quadruplexes. And I decided to train my models on intersection because it's more accurate. And for mouse, there is only one available G4-seq dataset, in vitro dataset, and there is no G4 ChIP-seq data for mouse now. Thus, our aim is to somehow annotate the mouse genome using human data. The first task was to build a model to predict quadruplexes formation on human data on the intersection of 2 datasets. And then we need to apply this model to mouse genome. And we need to somehow evaluate results because we don't have ground truth for mouse. We don't have G4 ChIP-seq data for mouse. And we have to develop some method to evaluate anyway. First, I preprocessed our data, human genome. It is a string of 80 GNCs, and so we need to one-hot encode it, transform it into vectors of zeros and ones, so our model can work with it. Then I split the genome into fragments, 5,000 nucleotides long. And this is our positive class, a class where there are quadruplexes on the genome. Now we need to choose fragments for negative class. Actually, the whole other genome is negative because there are no quadruplexes there. So we took random fragments, and our target is vectors of zeros and ones on each position. Zero means there is no quadruplex. One means there are a quadruplex on the position. And I chose an LSTM architecture for the model. This is a simple model of one B-LSTM layer. It is bidirectional because they work better on long sequences. Our sequences are long, they're 5,000 nucleotides. Then we have a linear layer that predict quadruplex formation on each position. And we trained it on the intersection of both data sets. And I evaluated some metrics for this. And on the right, we have examples of the model's predictions. Blue line mean predicted values, yellow line mean real values. On the top, we can see good predictions. On the bottom, we can see slightly less, slightly worse predictions. Actually, our data is unbalanced because there are no quadruplexes on the most of positions in fragments. So we have precision and recall about 0.6 and F1 score. And we have good accuracy. As we can see, there can be some errors on the edges of quadruplex. This is because of nature of ChIP-seq experiment, because ChIP-seq can make mistakes on the edges of fragment 2. And now we have this model trained on human, and somehow we need to apply it to mouse. This is an unsupervised domain adaptation task because we have a source domain is human genome, annotated human genome. So we have labels for this data set, and we have unannotated target domain is mouse genome, where we have to find quadruplexes. There are a lot of methods for domain adaptation, and I chose one of them. This is DANN method. It is domain adversarial training of neural networks. And to apply this method, we need to slightly modify our architecture. We still have BLSTM by LSTM layer that extract features out of data. We have again classifier. It is a linear layer which predict if there is quadruplex or there is no quadruplex. And we have a new additional block. This is discriminator block. It tries his aim to predict the domain of data on the input. And classifier tries to predict quadruplex formation, and discriminator tries to predict domain of model. And this network is trained in such a way that the future extractor has to extract features common to both domains. In this case, we could be able to apply it on human data and on mouse data because it extracts only common features. Let's look at little details. To train this model, we used common stochastic gradient descent method, as usually. And on each iteration, we have two steps. On the first step, we minimize the loss of domain classifier, loss of our discriminator. So we train our discriminator to predict domain, predict if it mouse data or human data. On the next step, we minimize the difference between the classifier loss and discriminator loss. So we train classifier to classify it to predict quadruplex, and we find it if discriminator could find out domain, if it can do it. It means we extracted some human-specific data from some human-specific features from data, and we have to prevent this situation. In the results, when it converges, we have a classifier that can classify successfully quadruplex formation, predict quadruplex formation in such a way that discriminator can tell if there is if it is from human data or mouse data. And now we need to somehow evaluate our results. We don't have ground truth for mouse. So I want to talk about this method. First, we ran a human LSTM model directly on mouse genome and had some annotation. Then we ran DNN classifier on the mouse genome. And we trained it on intersection of both datasets on human. And we have only one dataset for mouse. But if we trained it on the intersection, so found fragments on mouse data has to be in GFOSEC mouse dataset. And we counted the number of found fragments that are actually in GFOSEC dataset and compare it for both models. As we can see on the table, in case of LSTM on human data, trained on human data, there is same number of fragments that we can find in GFOSEC for mouse for LSTM and DANN LSTM model for both models, but we have a different total count of found fragments. We calculated the fraction of good fragments. If fragment is not in the GFOSEC dataset, it means this is a false positive results. And we can compare these fractions. And we can see that in case of D, A, and N, the fraction of false positive results decreased by 10%. So we can make a conclusion that we successfully adapted our model to mouse data. We have a smaller fraction of false results. It means we can use this method for in this task to train model on one organism's data and to adapt it to another organism's genome. So in this work, we trained an LSTM model, which is able to predict quadruplexes. We successfully adapted the model to mouse data using DA and N method, and we got a 10% false positive rate decrease. This is an improvement. That's all.
Thank you, Pavel. Dear colleagues, we have plenty of time. Please ask questions. Yes. Nikita will talk about prediction of secondary DNA structures. So please, Nikita, lead on.
Yeah, let's start. Hi everyone, my name is Nikita Konstantinovsky, and I'll tell you about generative adversarial network for DNA secondary structures prediction. Firstly, as already we mentioned, what is DNA secondary structures. So there is a lot of different DNA secondary structures, such as Z-DNA, quadruplexes, I-motifs, and other DNA secondary structures. Here in our work, we will apply our methods for G quadruplexes. Firstly, let's start from G quadruplex detection methods. There is several biochemical methods to detect G quadruplexes. As already Pavel said, is G4 ChIP-seq, G4 and G4-seq methods, and there is some computational based methods. It's like pattern-based methods, methods based on decision trees, and some neural network methods. However, there isn't some, still there is a way to improve these methods, because the accuracy is sometimes is not could be improved for these computational methods. First, I'll mention the canonical definition of quadruplex. It's like pattern-based method to detect quadruplexes. It's based just on a regular string, as you see on the screen. And this method detects around of 50% of all existing quadruplexes in existing data sets, and it could detect some some canonical structure of quadruplexes, but it couldn't detect some more complex quadruplex structures. So what about data sets? There is for humans, there is two two data sets. It's one was taken in vitro conditions. It's G4sec data set, and we have like around 700,000 number of G-quadruplexes. And G4 chipsec dataset, it's in vivo dataset. It contains around 9,000 quadruplexes. So we also, for other DNA secondary structures, there is even less data. For example, for Z-DNA, there is just 400 samples in data sets. Now about what we do with this data. What is generative models? Generative models is a way of learning any kind of data distributions using an unsupervised approach. All type of generative models aim to learn true data distribution of training set and generate new data from this distribution. This approach could be applied for data augmentation if we have lack of data, is as in our task of detection DNA secondary structure. Here we can see two major approaches for deep learning generative models. It's variation after encoders and generative adversarial network. In this work, we apply generative adversarial network. However, in process of research, we also try to use variation after encoders, and it shows less performance for this task. What about generative adversarial network? Let's talk about this, how it works. It exists from two neural networks, the generator and the discriminator. The goal of generator is to generate some real samples, some new samples from real data distribution based on some noise. And the discriminator aims to distinguish data between real data and generated data. So here we have, when we connect these two networks, it exists min-max gain between these networks, and so in the process of training, generated trains to generate data from real data, new data from real data distribution. We all know some applications of GANs. It's, for example, to generate some real people photos as on the picture. So these people never exist, but it generates some new people's face. Let's also look at some applications of generative adversarial network in genomics tasks. For example, here we can see the example of training GANs on two to generate exon coding sequence, and also we not only generate the exon coded sequence, but also generate the where intron stops and exon starts, the borders of exons. And after that, we can look at patterns which is in generated data, and we can see that GANs are trained to generate patterns of exon starting and exon stopping. So we can apply GANs for genomic tasks. Also, there is a lot of researches to apply GANs for data augmentation. Here, for example, some research which apply GAN for data augmentation on X-ray images of cancer, and they augmentate the data set, which consists of 400 images, and they augmentate it twice, and after that generated data with real data were passed to CNN based classifier, and its accuracy increased after this augmentation. So it proves that we can use GANs for data augmentation. Now let's talk about our approach. Firstly, we one-hot encoded original data, original quadruplex sequences. It's the same way as Nazar and Pavel said to you. After that, we test different GAN architectures for generator and discriminator, but the best one was CNN based residual network. Here we can see the residual block, and what is 1D convolution is what is based on CNN neural network of our approach. This network performs the best with other network architectures. Also, we should mention about generated data quality. It's a hard task to understand which quality of generated data is. Here we use different approaches, not just in train classifier for and look at the accuracy, but also we look at two metrics. We call it dself and dtrain. Dself is average edge distance in a set of generated data to nearest neighborhood within this set. It shows us that we generate different data, and dtrain is average edge distance in a set to the nearest neighbor in train set. It shows us that we generate different data from train set. So these two metrics which can show us the quality of data. Let's look at results. Here we can see metrics of dself and dtrain for real data and for generated data. For real train data, dtrain is of course 0 because metric because the distance between this dataset and train dataset is 0, but for other metrics we can see that resulted generated data is almost the same with real data, and it shows us that we in some case we can we have the same data, the data from the same distribution based on this metric. Also, we look at canonical G-quadruplex pattern in generated data set, and we can see that in G4 ChIP-seq, for example, how much, which percent of data is represented by this canonical G-quadruplex pattern, and we can see that percentage is almost the same. However, we can see that in 1000 generated sequences, there is more percent of extended pattern. It may show that we have some overfitting for this type of data, but it should be studied more deeply. However, we can understand that our generator could generate not only this canonical G-quadruplex pattern, but some more complex quadruplex patterns. Also, we compare existing G-quadruplex detection methods with our method. So what we do, the best method to detect G-quadruplexes is neural network based method. G4 detector it is deep convolution neural network, and on the right side we can see the AUX score for different negative class for different negative class for G4 detector, and also we train this G4 detector not only the real data, but on real and generated data and just on generated data. And here we can see that our score for real plus generated data, it's our data augmentation process, is more than for G4 detector just on real data. And also we can see that train classifier just on generated data also shows good performance in AUX score, so we can understand that our generator trained performed good. Also, we train not just generator which generates some quadruplex sequences, but also we train a generator which labels the borders of quadruplex in DNA sequence. So after that we can, if we shows the borders of quadruplex in DNA sequence, we can take the sequences of the same quadruplex lengths and look at the frequency in different positions for of frequencies of different nucleotides in different positions, and try to study some new quadruplex patterns based on this data. That's all. Thank you. You may ask questions.
Thank you, Nikita. Dear colleagues, please ask questions. We have plenty of time. We are ahead of the schedule. Okay, so no questions. Thank you once again, Nikita.
Thank you.
Next speaker is Alexander Fedorov. It's me. And I will talk a little bit about computational approaches for detection and quantification of RNA editing events. Yes. Now I will share my screen. Okay. We'll start with a small biological primer about RNA editing, then move on to the overall pipeline and data acquisition process for typical RNA editing study, then talk about published tools for the novel RNA editing detection, metrics for editing quantification, and I will finish with a few remarks and open problems in the field. Basically, RNA editing is core post-transcriptional mechanism of introducing changes to the RNA sequence. In a typical scenario, it looks like this. We have some RNA molecule and a protein, let's call it RNA editor. So RNA editor binds the sequence and introduces some changes, like substitution, for example, all adenosines converted to inosines, or deletions or insertions. Commonly, at least to the best of my knowledge, proteins RNA editors are capable of only one type of RNA editing, so it's either substitution or deletion or insertion. To give some perspective, the most common type of editing in, for example, humans is editing by substitution, and in particular, editing by introducing adenosine to inosine substitution or cytosine to uracil substitutions. They account for over 90% of all editing events in human transcriptome. Adenosine to inosine editing is done by ADA1 protein. In fact, ADA1 is structure specific, right? So it's a double-stranded RNA structure, and ADA1 binds such structures and destroys them. The second type of common editing, cytosine to uracil deamination, is done by ApoBec family of proteins, and it's in fact sequence specific. So ApoBec binds uracil adenosine rich sequences and deaminates cytosine to uracil. Okay, so these are two the most common types of editing. It's likely that you will observe these substitutions in real data sets and maybe focus on these substitutions. In terms of biology, these two types of editing are extremely important and involved in several cellular processes. For instance, right now we assume that ADA1 editing is in fact one way to distinguish host RNAs from pathogenic RNAs, though this editing is used as a mechanism for distinguishing host and self-RNAs by the immune system. So editing is not just a random anomaly in a cell, it's a real and significant biological event. With that in mind, let's move on to a typical overview of RNA editing study. Like many other biological projects, RNA editing is based on experiment called RNA-seq. It's a wet lab experiment. So we start with a sample of interest and extract and enrich our target RNAs. It is important to extract only RNAs you are interested in and enrich for RNAs, again, you are interested in. Because, for example, you want to study microRNAs, then by all means, enrich microRNAs in this step. Then these RNAs are fragmented and sequenced, as usually. Short reads Illumina sequencing. I highly recommend to use paired-end sequencing and as long reads as possible. Once you sequence your library, it's time for in silico or computational analysis. And first thing first is quality control of raw data and trimming low quality bases. So these are a few tools for this. And trimming is very important for RNA editing estimation because sequencing quality tends to degrade at the end of reads, and you don't want to bias your analysis by these low quality bases. Once you are confident in your raw data, you can align it to the genome using typical RNA-seq aligners like STAR or HiSET. And again, do next step of the quality control. First of all, assess your sequencing depths. A good study should aim for at least, well, approximately 80 million sequenced fragments, at least, I guess, 40, to get a reasonable level of editing estimation, a reasonable accuracy. And also pay attention to the library complexity. In fact, if your library includes lots of PCR duplicates, it's a bad thing because you started with a low amount of RNAs, and your editing estimation will be skewed by biases introduced by PCR during the amplification step. So the bottom line is that you should completely remove all PCR duplicates from your pool of reads if, say, the total amount of duplicates is like 30 or 40%, just not to skew the editing analysis. And once we are confident in our data, we can do de novo editing detection, we can calculate lots of indexes, we will talk about it a little bit later. One more important thing, technical thing regarding editing is that some reads after sequencing can be hyper-edited. It's especially important for adenosine to inosine editing, and it looks like this. Imagine this is a genome, and these are your A to I edited reads. And inosines in RNA molecules are read by sequencers as guanines, so A to G mismatches here are actually adenosine to inosine edits. So these RNA reads won't be aligned to this particular region simply because A to I edits account for a large number of mismatches. Right? So aligner won't consider this alignment as a good one. A typical workaround is to do a so-called RNA-DNA A to G conversion or 3-nucleotide alignment, when you simply convert all adenosines in the reference and in your sequence reads to guanines, and you don't have this problem anymore. These reads will be rescued. And it's especially important when you decide to focus on hyper-editing and to increase robustness in case you lack of reads, for example. So this is one workaround, but we also have HiSET 3 nucleotides. It's an alternative approach, a one-step approach. So I would recommend to focus on these two if you want to rescue these reads and focus on editing, hyper-editing in particular. Okay, now an important and interesting topic, de novo editing detection. We have our reads, and it's basically core data for editing estimation and detection. And here is a short table with summary of RNA editing tools. In fact, GRME, VS Scanner, and RNA editor are no longer supported. And for new studies, I would highly recommend to use either JACUZA or RediTools. They both support RNA-seq data alone. They support stranded RNA-seq, which is also important in the context of RNA editing estimation. And they also can compare your RNA-seq and DNA-seq, if you have any, to remove all false positive hits. Here I should know that actual filtering, right, selecting a set of true editing events from separating true events from false events, is usually done using a set of heuristic filters, and results strongly depends on parameters you used in these heuristic filters. So I want to just give a few examples and an intuition behind the common filters which you can find in JACUZA or RediTools. First, it's a set of filters associated with a particular site. So it's mean coverage per site, mean editing frequency, and mean number of reads with a given editing type, and mean PRED score It's a score associated with each position in read. So you don't want to pay attention to reads which were sequenced with a low confidence, right? These thresholds, like mean coverage more than 10 reads, for example, these filters are used to reduce your noise, sequencing noise, increase reliability, and in fact, increase your precision, create precision for recall. So you have more confident hits, but less hits in general. Second very important thing is to remove sites in homopolymeric or simple repeats. These regions of the genome are extremely hard to sequence correctly and align correctly. So these two are the common source of false positive editing hits. And it's also important to exclude position where we observe multiple editing events like A to G, then G to A and T to C, for example. These sites are either prone to sequencing errors or these are special assembly anomalies. For example, imagine a genome, real genome, and you have two almost identical regions in the genome, which are both transcribed. But in the assembly, you have only one region, and RNAs from both regions will align to one region in the assembly. But these two regions in the genome can be a little bit different. And that's why after the alignment, you will have strange editing pattern, which is actually an anomaly, an assembly anomaly. It's better to remove these sites from the analysis completely. It's also a good idea to exclude genomic SNPs, but only those that are not based on cDNA libraries, coding DNA libraries. So those that are not based on RNAs, only on DNAs. I already talked about read streaming. It's also important. And one thing is to maybe you will consider an essential to avoid reads mapped with a low quality, with a low confidence. So you can introduce a min mapping score threshold or exclude multi-mapped reads. It in general could reduce your alignment errors, but still it's more to the target of your study. Right? So multi-mappers can be biologically meaningful. For example, you have multiple almost identical genomic repeats in the genome, and they can align to both of them. So you cannot, aligner cannot resolve them. But once you manually check these hits, you will see that one repeat is in the middle of nowhere and one is in the 3' UTR. So you will discard this edits and keep this one. And one more thing, please, if you have a low complexity library with lots of duplicates, exclude duplicated reads completely. Otherwise, they will skew your editing estimation. One more thing to note here is that it's fairly common to check weak hits or vice versa strong hits in public databases. For such purpose, I usually refer to the READY portal. It's the only large-scale RNA editing database maintained today. Especially, it's a good source for human RNA-seq editing in humans. It contains like 10,000 RNA-seq samples. Unfortunately, a lot less for mice, but still, it's the only database available today. Maybe you will encounter DARNet or Radar, but well, they are not available anymore, unfortunately. Okay, now we have a few tools to discover editing events de novo. Now let's move on to metrics, which can be used for editing identification. And the first simple disclaimer, every formula on the next slides will be provided for adenosine to inosine editing. Remember, A to G mismatches, because inosines are read as guanines. But all these ideas readily translates to other types of editing, so it's not a big problem, I think. The simplest metric is editing frequency. So you have a particular site in the genome, and you can calculate editing frequency, observed editing frequency. Number of mismatches divided by the total coverage. It's a localized metric, you know, a particular position in the genome. It's fairly easy to understand and calculate, and given an extremely deep sequencing depth, it's the best editing measure for a particular site. However, it's not usually the case. You don't have millions of reads aligned to a single site. However, it's also the only your, let's say, choice when you want to select sites for experimental validation. So you usually want to select sites with a sufficient coverage and the highest editing frequency. The main disadvantage of this metric is that we observe extremely high variance for editing frequency in particular sites due to coverage, and variance is actually originates from the coverage difference, library complexity differences, differences in protocols used to prepare libraries, even in data preprocessing. Editing frequency can vary based on the aligner you choose during the alignment step. Okay, so keep this in mind. To decrease possible variance but still keep a localization as a feature, we can focus on feature editing index. In fact, we need just to select a set of features of interest, like 3' UTR of a gene for each gene of interest, or introns of genes, or something like that, and build a set of reliable editing sites in each feature. So for genes in each 3' UTR, for example, or in all exons of each gene. Then feature editing index is simply a total number of mismatches for all sites in a given feature divided by the total coverage of sites for a given feature. So this index is calculated for each feature we have. So for all genes, for all introns, or something like that. It's a localized metric, which is good. We can relate it back to a particular region genome, to a particular gene. And actually, it allows the way we formulated this allows us to trade off localization, signal-to-noise ratio, and interpretability for lower variance. So the more sites you include in this metric, the lower the variance, and unfortunately, the lower the interpretability and maybe the signal-to-noise ratio. Right? So more sites, lower variance, less sites, higher variance, but also a few things to consider about interpretability and signal-to-noise. In extreme cases, this index can be driven by highly covered sites, but it's usually not the case if sites are close to each other. And again, reliability depends on many factors like coverage, number of sites, library complexity, data preprocessing. Still a good tool to use in real studies. One of genome-wide measures for editing is a so-called overall editing level. We start with a set of reliable editing sites for the whole genome, not for some features, for the whole genome. We can derive them using de novo tools or use all sites from a public database like ReadyPortal. And then the index, the editing level itself is just number of mismatches summed across all sites divided by the total coverage summed across all sites. The good fact is that variance associated with this overall editing level measure decreases rapidly, at least for adenosine to adenosine substitutions. So we reach a plateau at approximately 100,000 sites, which is not that much for human. We have millions of editing sites in humans. However, again, it's possibly sensitive to outliers where you have a single site, extremely covered single site, then this overall editing level will be skewed because sites are pooled with an average across sites. And it's also, yeah, the number of reference sites is small, it can become, well, variance will be high. Remember about this. One more important or say interesting index you can consider to analyze is a so-called editing index, recording editing index. Imagine a gene and a few editing events which happen in coding sequence. And actually editing leads to non-synonymous codon change. For example, here A to A is easily seen, and after editing we have A to G, which is methionine. And the idea that we can collect all such sites across the genome, across all genes, sites where editing changes the codon, and again divide mismatches by the total coverage. At least for adenosine editing, it's a good proxy for AD2 activity. And again, it's a good proxy for a good genome-wide editing measure. Unfortunately, it's quite unstable with low coverage, and again, might be dominated by highly covered sites. There is one more index called allele editing index. It's adenosine editing specific, but unfortunately, I ran out of time, so please refer to this publication if you're interested in details. In conclusion, editing, RNA editing is a trending topic. And what is good for us as researchers is that we have a lot of questions unsolved, yet unsolved. And please consider maybe you'll be the one who will solve. For example, you have ideas how to design and implement a framework for differential RNA editing analysis. Those current solutions, well, underpowered, to say the least. Thank you very much for your attention. I'm ready to answer questions, if any. The next speaker is Dmitry Konovalov. He will talk about zRNA formation in cells infected with virus. So please, Dmitry, lead on. Dmitry, sorry, we can't hear you.
Oh, sorry.
Yeah.
I will repeat introduction. So, my name is Dmitry Konovalov, and today I will present a short report about zRNA induction, the response to viral infection, and the most focus would be on IEV or influenza virus type A. It's known as flu. And begin with a reminder what is zRNA. So typically RNA has A form, which is right-handed information, and under some conditions it can become zRNA, which is left-handed form, and you can see it on the picture. On the left, it's RNA, and on the right, it's zRNA. And under infection, it was observed that for some viruses, not all of them, only some, we can see that after infection, zRNA is formed. For such viruses are HSV or herpes virus, IEV, influenza virus, IPV, bronchitis virus. And in this picture, you can see that some viruses like rhabdovirus or poliovirus, they don't form, don't induce zRNA after infection. And it's not really clear why this behavior is observed. There are some ideas because this group of rotamyxoviruses which includes IEV, has some special feature, and this feature is replication in the nucleus. But there are some evidence that it's not required, because RNA was detected in the cytosol, not in the nucleus, so it's not required. Also, ZBP1, the protein which is the main protein that recognizes IEV infection, and it's still active when it's excluded outside of the nucleus. So it's also good evidence that maybe nuclear replication is not a thing that's required. Also, it was observed that ZBP1 senses mitochondrial damage. Also, that the alpha domain is a domain which binds to the DNA and the RNA, and this domain can work in the cytoplasm. And also some viruses, such as pox viruses, they itself have the alpha binding domain in their genome. So there is a lot of things to determine, and the things that require further investigation. Why some viruses induce the RNA formation, and why others don't. And we'll talk about influenza virus in details, because it's really common. Over 1 million people annually are infected by influenza, and influenza is a virus which induces the RNA formation, and there is hope that gaining some knowledge about this process will help to cure some clinical cases, because these pathways, which include ZBP1 signaling, result in cell death, and uncontrolled cell death can lead to some severe results, such as pneumonia and mortality. That's the things I think that all of us want to prevent. So when we look at RNA formation in IEV, it was discovered by our colleagues, Tinjan, that influenza triggers RIPK3 pathway, and if pathway works through RNA binding by ZBP1, which results in RIPK3 mediated necroptosis. And the simple scheme of this pathway is shown on the right. So on the top, you can see the virus. It induces some RNA formation. ZBP1 binds to this RNA. After that, ZBP1 activates RIPK3, necroptosis pathway or apoptosis pathway, because there are multiple ways of cell death, and which one would be active is a result of several pathways, and libk3.1 can be responsible for apoptosis and microapoptosis. But it's not only one way of cell death, because there are other ways, for example, pyroptosis, which is not mentioned here. And this is, it's not covered in this discussion. So when we look at AAV, and we can try to check if this RNA information, if it already was there, and we can see that indeed the RNA is formed after infection. On the picture, we can see the RNA shown in red increases dramatically over time after infection. So as 12 hours, we can see that it destroys nuclear membrane, and cell is dead, and it has lots of RNA. And this amount of RNA is correlated with rate of virus replication, and also it's proportionate to amount of virus inoculum. So more viral particles, more RNAs in result. And the question is, what RNAs are RNAs? And there are some potential candidates, for example, some nuclear RNAs. There are some mechanisms which would, for example, after infection, some editing might occur of nuclear RNA, which will change its formation to RNA. Also, infection typically changes some transcript usage and changes replication, which can result in some stress that will form the DNA and the RNA in one RNAs. Also, it can be a viral RNA because viral particles are packed, and this packaging can induce some torsional stress, which will give enough energy to form the RNA forms. And also, one of the candidates is defective viral genomes or defective interfering particles. And it was shown that when we have lots of these particles, we have lots of RNA. You can see on the picture on the right that IAV-LD, it's a cell with low amount of these particles after 6 hours post-infection, and there is not much of RNA. In comparison, on the right, IAV-HD, high rate of these particles, and lots of RNA. And to make this clear, why we have RNA and where we have RNA, we need an experiment, and this experiment was already performed with the following design. So we can take some cells, for example, mouse embryonic fibroblasts, which are modified with flag ZBP1 for localization of ZBP1. And these cells we perform 4 experiments under the cells. The first one is Z22, the main one, and Z22 is an antibody for the RNA and the DNA. So when we perform RNA immunoprecipitation sequencing or RIP-seq with these antibodies, we can get RNAs which are the formers they would be pulled down by these antibodies. But to properly evaluate these results, we need some set of controls, and our 3 experiments are these controls. The first one is input, it's control for expression, and it's total RNA-seq, which results in whole genome RNA expression profile that can be compared to Z22 expression pulled out. And IgG is a control for specificity because our binding can be non-specific, and we need to control for this effect. But unfortunately, IgG is kind of problematic control because it has its own biases. There are some problems with normalization of this data, and mostly we rely on input control. And J2 control is control for double-stranded RNA. These are antibodies which bind to double-stranded RNAs. And for all these sets of experiments, we perform them for 3 time points: before infection, 8 hours after infection, and 12 hours after infection, to check for dynamical changes in expression of the RNA-forming genes. And these raw data obtained through these experiments require some analysis, and we need to trim these reads, align them to genome, remove ribosomal RNA, because protocols of ribosomal RNA depletions are not perfect, and we need to do it anyway, even if ribosomal depletion was performed by the lab. After that, we want to quantify our reads. I did it by Selman. And after that, we want to check the differential expression, for example, by dissect2. And after that, we want to see our list of genes which are differentially expressed, and perform pathway analysis to check which pathways are involved in, which pathways include those genes and were most affected by RNA formation. And also, we want to check some editing, which I believe Alex described in great detail in his talk. And what about results of this analysis? They are not published yet, so I would not go into details. But in brief, we observed enrichment of mitochondrial genes in Z22 pull-downs. We observed that over 300 Z-forming genes are activated after infection, and those genes are enriched in interleukin 17 signaling pathway, NFKB pathway, and not surprisingly, influenza A pathway. And also, right now we are waiting for some experimental data with deeper sequencing, and we believe that those data will help us to improve our analysis, because sequencing depth and quality of RNA purification, they add, when they are not done properly, we have lots of noise, and also we have lots of problems with RNA editing detection, and these changes in lab will greatly increase the quality of analysis and results. And I believe that's all that I wanted to present on this topic today. And I'm ready to answer any questions if you have ones.
And I'm happy to start our second part of this session, of the bioinformatics session, this big conference, and present you Dr. Professor Alan Herbert, who is a founder and the president of the company Inside Out Beer, sorry for the confusion of the name, based in Boston, USA. And luckily we have him as a scientific advisor of our International Bioinformatics Lab at HSE, and hopefully soon as the pandemic is over, we see Professor Alan Herbert here in the walls of our campus on Pokrovsky Boulevard in Moscow. But so far we have this Zoom communication, and please, Alan, have your word. You can share the screen as we did.
Thanks, Maria. So can you all see that?
Yes, we do. Just
okay,
it's not full screen. Oh, now it is. Mm-hmm.
Okay, well, thank you very much, Maria, for the introduction, and thank you very much, Sasha, for the opportunity to speak at Max Pro 2021. I've listened to some of the talks this morning, and they've been really great, and I'd like to thank Sasha and Dimitri for warming up the audience to things left-handed. So today we're going to talk about a new form of genetic encoding that we've been working on for the last few years, but now we have a lot of biological validation for. So the traditional idea of how information flows in cells is from the genotype, it gets processed into RNAs that contain codons that encode proteins that produce phenotypes. And the fun thing about multicellular organisms is there's just not one genotype. There's like thousands of genotypes potentially buried inside the genome, and it depends on which particular genotype you extract as to what the consequences are. So, it's an interesting system where when people say, are you going to get your genome sequenced? Well, that's fine, but each cell actually takes a different slice of that genome to produce outcomes that vary across tissues and across individuals. And so this is the classical view of how it works, and there's a few exceptions along the way where there's feedback from protein to how RNA is processed or how DNA is read out, or even from RNA to DNA. So, the classical paradigm has survived since the 1950s with a few kind of interesting exceptions. So what we are going to be talking about today is slightly a different way of thinking about how the genome works, and sort of, in a way, it's analogous to the way we divide computer into software and hardware. So, what I just talked about was all the codon that encodes proteins, and that gets translated in the ribosomes in the cells to produce machines that constitute the wetware of the cell. What we're going to be talking about is something called flip-onware, which is really a way of compiling codons to make different proteins to produce different outcomes. So we're going to be focusing on this kind of compilation that's going on at the genome level, and also occurs at RNA, but for the purposes of the discussion today, we're just going to think about how flip-ons affect phenotypes and how they change outcomes, so that inputs, the same input can be processed in very different ways. And depending on the context, you get a variability in output. So we're going to start like imagining that Dimitri and Sasha, who gave a really nice presentation earlier on, applying for a job, and they applying for a job as a computer programmer. And so we're going to, so the interviewer says, Alex, Dimitri, what have you got to show? And they said, well, we've come up with this new programming system. It's self-replicating. That's great. It's self-repairing, so it's really resilient to damage. That's excellent. The code base is really expansive. We can add stuff in there and we can combine them and get lots of interesting programs produced really easily. And then we can select which program that we need to do the job. And by this time, the interviewer is getting really excited. It's also, Alex and Dimitri might say, really redundant. You know, we can be sure that we can get a particular class of responses from it, depending on how we select it. And by this time, the interview is about to give Alex and Sasha a huge check to come and join the company. But then they mentioned that this whole program they've developed is really expensive. It makes lots of errors. And actually, most of the output from that whole system is just trash. It's just junk and needs to get rid of. So there's just a whole lot of overhead and cleaning out all these mistakes. And finally, they say, well, and this is why they didn't get the job, you can run this program 1,000,000 times, but you never get the same result twice. And that's kind of the difference between the way we think about computers and the way that biology operates, that what we would do in terms of the computer code being clean and efficient and lean is exactly not what we find in biology. So we need to understand that more, because clearly the biological programs are highly successful. So let's just go into those little categories a little bit more. By expansive, what it means is that you can take any sequence of DNA, throw it into cells, and then you can change their function. You can get a gain of function, a loss of function, or actually you can make a whole new set of functions depending on what code you put in there. So imagine writing a program where you just send in a string of the alphabet there, and suddenly you can do all these new functions. You know, just throw it in there, and then you're able to select some combination of those letters you feed in there, and suddenly you have a whole new program. So what's more is that once you do that, and throw in those string of letters and code and all that kind of stuff, what's really interesting is that you're not limited to what you throw in. There's a whole system of combinational diversity that's generated. And that's in multicellular organisms due to the fact that the genes are encoded in pieces. So the functional part of a gene may be, in this case, in 3 separate pieces. And to actually get something that makes protein that actually works in the cell, you need to combine these pieces of the gene together and splice them. So you can combine these different functional elements that are called exons in lots of different ways and produce lots of different RNAs. And some of these are functional, some of these aren't functional, but they all lead to different protein products. So it's expensive approach, because actually most of these RNAs that get spliced actually don't do anything. For example, you have to get rid of all these introns. They're not necessarily functional, so you make them, and introns can be up to 40% of the genome, and you make 40% of the genome into RNA, and then you just trash it. It's not doing anything for you, so you get rid of it. So that's expensive. And also you make lots of mistakes. You can join these exons together in a way that they don't make sense. And they're not going to do anything except introduce errors into the cell. So you got to get rid of them. And there's a whole cellular process called nonsense-mediated decay that gets rid of all this junk RNA that you make. So it's good in terms of combinatorial diversity, but it's bad in terms of the energy it takes to make sure that you don't end up introducing errors into the cell that end up destroying it. Well, it's also kind of expensive as well in the sense that you can make lots of different types of cells with all this combinatorial diversity. And here's just 4 different cells that are going, 2 are going one direction and 2 are going in another direction. They interact. And what's indicated here by these little T-shaped arrows is that each of these cells are capable of killing each other. So in fact, the way to get the response that you want involves getting rid of a lot of cells. You kind of have to kill these 2 cells here if you want to have a repressive response. And this is from the immune system. These are repressor cells. These are natural killer cells. So to get a repressive response, you need to kill off a bunch of cells. And to get a cytotoxic response, you have to do the opposite. You have to kill off the other pair of cells. And it's a kind of an intransitive logic that's involved where whichever cell predominates is determined by the context. So you can use the same cells, and by killing off some, you can change the nature of the response in a context-specific way. And again, that takes energy, it's expensive. You're making cells just to kill them because they aren't going to serve any useful function in that context. Also, it's kind of selective as well. Of all the potential RNAs you can generate and all the potential responses that you can make, you only want to select those that are relevant to the context. And one of the biggest challenges is that when in multicellulated organisms that have 2 parents, you have to make sure that these genes from the different parents get along because they've never seen each other before and they don't know what each is capable of. And so you can't let one parent dominate the other parent because of the potential issues of incompatibility between the gene products. And you can just kill the organism, the embryo, at a very early stage. So you need to be able to select which of those RNAs get generated and which of those get expressed as proteins. And you can change the threshold depending on the context as well. So it's a system that makes lots of RNAs. It's combinatorial, but then the RNAs can come from anywhere. So you have to have a lot of controls and a lot of thresholds to control your responses in a context-specific way. So as I said before, when we're talking about the cells, most of the logic inside cells is intransitive. So as computer programmers, we always think of, A to B to C to E, and that if we have A, then therefore we get E. So we always think in transitive logic, and it's good for science because it means results are reproducible. It's confusing for quantum mechanics because you can have states that are incompatible superimposed with each other. But anyway, you can kind of get around that by making some additional assumptions. But actually, biology doesn't care much for transitive logic. Most of the responses are intransitive, where in this situation, E can actually dominate A. So there's a game called Rock, Paper, and Scissors, which kind of illustrates intransitive logic, where you have 2 players that make a hands symbol, either for a rock, a paper, or a scissors. And so rock will beat scissors, scissors will beat paper, and paper will beat rock. So depending on what the other player plays, one response will predominate, another responses won't. The other advantage of the intransitive system is that you can have inputs at any level that can produce outputs at any other level, where in the transitive system, you don't get such flexibility. So this transitive system actually limits variability, which makes it programmable. And that's why we like it, because we can control the variability. But on this system here, it's built for generating diversity. So it's a different logic that's present in cells. And if you want another concrete example of it, you can think about the Krebs cycle, for example, where the carbon backbone gets passed around and around and around, and different inputs come into the Krebs cycles, and there are different outputs. And depending on the context, one input will be important, and in another context, a particular output will be important. So that's kind of captured in this slide, where not only can you input at any of these points, but you can also output a lot of these different points. And so from a biological point of view and from a computational point of view, it's really interesting. So if you actually treat these symbols here as numbers, so like in the usual sense, so from a number system like 3, 4, 5, 6, 7 in an ordered fashion, you can see that we have numbers that aren't sequential. And so from a computational point of view, that's great, because then we can make conditional logic. So we can say we will only get 5 produced if we have 2 coming in from some other source. So we can make this whole step here conditional. We can also build in redundancy into the system, where we can, if we want 4 to go to 7, we can either have 2 or 3 add together to give 9, allowing us to do that, or else we can just input a number greater than 4 into the system to get that step made. And the other thing that's fun for a biological system to do is that it can actually bypass many of these steps. So if under some condition of selection that it's optimal to go from 6 to 4 and avoid going through steps 3 to 5, you can select that process. So it's a very flexible system in terms of the logic of biological cells that we don't really capture in terms of the way that we program computationally. So a lot of this redundancy that results from this kind of cycle is actually built into biological processes. And it's, anyone who's sat through any bioinformatic large data set analysis will normally pull their hair out, because all they see are tangles of interactions and pathways, and it's really hard from a transitive point of view to try and understand what's going on. It's really difficult to kind of connect the arrows in a way that a transitive person thinks makes sense. In fact, a lot of the biological interactions are intransitive, and there are many paths from A to B, and this redundancy that's built in is based on a lot of very low affinity interactions that can arise over time and be selected. And it allows a cell to be very resilient to environmental changes, and also really resilient to errors that occur in the DNA code or the introduction of new DNA code into the cell. And I think for those who are following recent developments and thinking about how gene transcription is regulated, people are now thinking, rather than having specific transitive interactions between DNA and proteins, you actually get a lot of these condensates form, where there are a lot of low affinity interactions between cell components that contribute to A, leading to production of B. So because of all these kind of different paths that exist in the cell from getting from A to B, it tends to make the system very canalized. So it tends to, if you imagine this ball rolling down at the top of the mountain, it has a choice to go between these different valleys. And that's the only choice it really has. It can go A or it can go to B. But there's a whole other space it will never enter, because all these redundant, resilient interactions will guide it one way or the other. And this concept, was developed in the 50s by this guy called Waddington, and it's called canalization of phenotypes. So despite all the diversity that you can generate, one way of handling it is to build in redundancy and resiliency so that only certain outcomes will happen. And in terms of self-replicating, well, depending on which way you want to look at this, whether this guy is running away from a lion or this guy is running towards a potential reproductive partner, what happens is this selection for variation, so that survival of a species by propagating the next generation is something that is favored by natural selection. And along that process, variation accumulates from all the errors in the genome to make this process more efficient. So self-replicating computers is not something that we can make currently, but biology provides a grand example of the benefits of having a self-replicating computational system. So talking about how all this information is encoded in DNA, traditionally we talk about codons, and codons really represent the order of nucleotides in the DNA string. And so we have something that's called a DNA code based on codons, but it's actually a cipher, because it just maps nucleotide triplets to amino acids. And it's just a lot of strings that are slapped together that enable you to make functional wetware. what we're going to be talking about today are flip-ons, and rather than the strings of DNA, we're talking about sequences that adopt alternative conformations. And I'll give you some examples in just a moment of what an alternative DNA conformation looks like. And what the current way we're thinking about that is that this encoding by shape actually provides an instruction code for compiling transcripts from the genome. So in this combinatorial process that I mentioned before, flip-ons can actually help decide which exons compile together to make a message. And so they can change the output from the genome. And because their shape is variable, depending on the context, then you can get different products from the same gene just by changing the DNA shape. So the advantages of codons, of course, is that because it's a combinatorial encoding by 4 nucleotides, you can get really high specificity in recognition of proteins and in terms of control. So you can give everything a distinct nucleotide barcode, for example, and you'll be able to specify it with high accuracy. The other consequence of using nucleotides is that you can introduce variation by mutation or some other process, and then the outcomes will change depending on what the changes that you make. The disadvantage of it, if you're going to have high specificity by having lots of variation in sequence, then getting a search efficiency going to find that particular sequence varies with genome size. And so the larger the genome, the larger the sequence block you need to be able to ensure that it's specific for a particular outcome. It's also another problem with encoding with nucleotides is that many different proteins and many different sequences are repeated within the genome, and each of these sequences or these binding specificities compete with each other. And so actually trying to find that particular sequence that you have generated with high specificity can be really challenging because you have a lot of noise from related sequences and overlapping motifs that make the search less efficient. In contrast, and all these different operations that you can do with nucleotides are described by string operations. And in biology, most of these string operations are usually one-way functions because it's actually more efficient to do that. You know, by a one-way function, I mean that you can't go backwards. To revert back to the original sequence is kind of really challenging in a biological context. Most of the mutations that occur are imprecise. So having a mutation that changes the sequence and trying to find the exact mutation to reverse the change is pretty difficult to do. So rather than trying to go backwards, the biological solution is just to get rid of those cells where you generate errors in the DNA, in the string, that you can't fix. And so you basically use cell replacement to allow a system to reset. So you go back to the original genome in a new cell and start again. So most of the string operations that are done are pretty well characterized now. You know, there's clearly DNA mutation, recombination, repair pathways that are error prone. There's DNA editing, much similar to the RNA editing that Sasha described. And there's also DNA transposition. So generating new strings can, within a genome, can occur by the transposition quite frequently. And that's the most common way that new strings are introduced into the genome, is having one segment of DNA transcribed into RNA, and then reverse transcribed into DNA, and then inserted somewhere else back into the genome. And so in humans, that accounts for about 50% of the origin of our genome, where we've had spread from one locus to another locus through this RNA intermediate and through this reverse transcription process. In other organisms, you can go directly from one locus to the other locus just by copying the DNA from locus C and inserting it in locus D. So that's when you're talking about string operations, that's an example of one type of string operation to increase the coding base for your DNA. As Sasha also introduced, there's other ways of entry of, there are other one-way string operations, and these don't act at the level of DNA, they act at RNA. So you can change the nucleotides that are present in the message by the process of RNA editing. And it can be quite extensive, like you can have addition, deletion, editing, where you get insertion or deletion of bases. And this is best well known in the trypanosomes, where their genome is completely non-functional. It's completely trash. If you read out the sequences from the DNA and try and translate them into protein, all you get is a bunch of stop codons. You can't make a protein from a trypanosome mitochondrial DNA genome. In fact, the only way you can make functional proteins is to insert or delete uridines. And so that's kind of the biggest contrast I know of, showing the difference between the way things are encoded in biology and the way you would encode them using a computer code. The other type of editing which is found in humans is the substitution editing, where you can get a change in one of the adducts on the ring, in this case from an amino group to a keto group. And so in this case, you change cytosine to uracil. And you can also do this for double-stranded RNA, which we'll talk about later, where you get the amino group removed from adenine and you end up getting inosine, which is translated as guanine if it's in a codon. So these are just examples of string operations that happen on nucleotide sequence. So in contrast with flip-ons, flip-on-based logic is a lot less complicated and operations are a lot more limited, but it's a much more powerful system just because of these limitations. So there are only very few number of structures that DNA can adopt, and nature seems to take advantage of that. And so you're either in one structure or the other structure. So most of the outcomes are binary. So rather than having some kind of analog variation that you can get by varying the nucleotide sequence and varying the protein that's produced, you can just produce a 0-1 outcome. It's either bDNA or it's in some other conformation. And again, you can also have some elaboration on this scheme because some sequences can adopt multiple alternative conformations. And so you can get competition between whether bDNA flips to Z-DNA or flips to a G quartet or some other conformation. And we'll talk about that in just a moment. And so rather than talking about string operations, what we're talking about with flip-ons and structure encoding is transition probabilities, whether basically it's a measure of how hard it is to flip from B to some other conformation. And the other difference that we have between flip-ons and codons is the whole process is easily reversible. So you can go from B to something else and back to B again. And so in that process, you can get a lot of heuristics involved where the forward and reverse rates are quite different. And you can actually vary the rates by varying the modifications that happen to the DNA. So we'll talk about that in just a moment. So here you go. This is the bDNA chromosome here, and here's some example of flip-ons where you can flip from B to Z and just by inverting the bases. And so you go from right-handed to left-handed. You can refold the DNA in a way so that's a 4-stranded structure. And this happens in G-rich sequences. And so you end up getting what's called a quadruplex. You can actually also take one DNA strand and anneal it to a bDNA helix. You can put it into one of the grooves and end up with a 3-stranded structure called a triplex. And also you can do other fancy things by varying the sequence here. You can actually introduce bends into the DNA that actually restrict its topological properties. So there's no shortage of alternative conformations that DNA can adopt. And the question always has been, what does nature do with this potential diversity and this potential variation? So here's some potential roles for these different conformations that nature can use them for. So with Z-DNA, clearly it's a binary switch, B to Z to B to Z to B to Z. You can switch on and off a number of things, and I'll give you some examples later on. these G4 quartets are really stable. So they form rapidly, but it's hard to reverse the process. So potentially there are memory elements. There are other conformations that we didn't mention that pH dependent, so it could act as the pH sensor. We have the T flip-ons or the triplexes incorporating this third strand. This third strand could be an RNA, so it could actually end up as a way of integrating output from a gene with the actual state of that particular gene. So if you form a triplex, maybe that's a way to indicate to the gene that it's in an active state and to maintain that active state. So these bent DNAs can act as insulators to stop topological strain from being transmitted through DNA. And then we have bDNA, which by comparison is just rich in nucleotide variation, but is not nearly as dynamic as all these other alternative structures. So we're going to actually talk mostly about Z-DNA, and again, the transition is reversible and it involves just flipping the bases over, inverting the bases, and in the process, you end up changing the direction of the backbone. So instead of going from being right-handed, going from right to left, shown by the yellow strand, it's left-handed with the backbone going from left to right, as shown by the red strand. So that's the difference between B and Z-DNA. It's just an inversion of base pairs. And so what we're going to talk about is the physical nature of this flip, what causes it to happen, the biological implications of being able to flip from B to Z-DNA in a binary manner, and also the computational implications of having a flip-on like Z-DNA. So Z-DNA is, like many of these flip-ons, actually occurs mostly in simple repeat sequences where you have a preferred sequence that will go from B to Z. And for example, in this sequence where you have alternating C and G, why that is preferred is because the Z-DNA actually has a dinucleotide repeat in the backbone. The dinucleotide repeat is associated with the structure of the backbone, and we'll talk about that in just a moment. There's also other sequences that are repetitive, such as the allo sequences that were copied through RNA from one position in the genome to another position in the genome, and they're about 12% of our genome. So they also have Z-forming elements. And also endogenous viruses end up forming Z-DNA or Z-RNA just because of all the topological problems they have in packaging themselves into protein capsules. So the interesting thing about all these repeat sequences is that they can be spread through the genome relatively easily, either through recombination or through reverse transcription. And the effects on the readout information at the new location is completely is locality specific. So they change the readout at that particular location. So you can spread these flip-on sequences throughout the genome, and then you can select the particular flip-ons, which changes the readout from that locality to give you a beneficial phenotype that can be selected by natural selection. So they're very active elements of the genome, changing shape. They spread around the genome, and they're also genetic elements because they are subject to selection because they affect phenotype. So part of the physics of, or physical chemistry of B to Z transition. So Z-DNA is a higher energy state. It takes energy to flip the bases over and to form Z-DNA. And as I said before, it's a transition that doesn't normally follow the same forward and backwards pathway. So it can takes the time to form Z-DNA versus the time to relax Z-DNA is different because of this ketosis that occurs here. So there are different things that can affect the energetics of this transition. So you can put base adducts there that change the propensity for bases to want to flip over. And you can also put energy in there in a number of ways. And one way is supercoiling, and we'll describe that in the next couple of slides. So here are potential base modifications that can occur on the rings. So when we talk about adducts, we're talking about modifying like cytosine or guanine, for example. And there's a whole variety of different modifications that occur, which can increase the propensity to form Z-DNA by lowering the transition energy. And amongst these well-known 5-methylcytosine and all the oxidative derivatives of 5-methylcytosine that also affect the potential of bDNA to flip to zDNA. In terms of guanine, we can get oxidative modifications such as that affect the ring position, and they also increase the propensity to form zDNA and also G4 DNA. So in some ways, in those particular contexts, these base modifications allow a cell to sense that there's oxidative damage occurring and form an alternative conformation that can activate particular biological repair processes. So in terms of supercoiling, this is something that happens in covalently closed circles of DNA. So if you imagine this is a double-stranded RNA like a plasmid, and it's covalently closed, there are no gaps or anything like this. And so it's twisted around itself a little bit. And so one way to relieve this twist is actually by taking a segment, the segment of bDNA and flipping it to zDNA. So you go from right-handed bDNA to left-handed DNA, and you absorb these writhes and relax the plasmid. So that provides energy. The energy of the supercoiling provides energy to stabilize the zDNA conformation. And what's kind of interesting, and you'll see why in a short time, is it takes about 1.65 negative supercoils to flip this bDNA into zDNA. And about 1.6 of that is the actual difference in the helical parameters of bDNA and zDNA. But also you have to allow for the fact that there has to be BZ junctions, the junction between left-handed and right-handed DNA that causes some unwinding as well. So as I mentioned, switching the shape from bDNA to zDNA requires power. One source of that power is RNA polymerase. As it goes down the double-stranded DNA, it unwinds the double-stranded DNA so it can make RNA, and that unwinding causes negative supercoiling, which can power the flip from bDNA to zDNA. Another way of doing it is actually by stretching DNA. So if you have a helicase that stretches the DNA, so it pulls on the end, that also introduces tension in the DNA, and you can relax that tension by flipping from B to zDNA. And the reason the tension is relaxed is because bDNA is 34 angstroms long and zDNA is 45.6 angstroms long. And so by doing the flip, you can make the DNA longer. And so when you try to stretch it, flipping it to zDNA relaxes the tension that you generate. And this can be shown with single molecule measurements with high degree of accuracy, and all that energetics is well quantitated. The other interesting way of doing it is normally our DNA is wrapped up around nucleosomes, and that allows it to be really compact. But if you flip out the nucleosome and just lift with DNA, you can then generate these negative supercoiling, because the negative supercoiling is constrained by this solenoidal wrapping. But when you flip out the nucleosome, suddenly you have all this negative supercoiling generated, and then that can provide the energy to stabilize zDNA. So there's a variety of different ways that you can flip a sequence from B to Z. So that's all fine in theory, but what about the biology of zDNA? We have all these nice physical chemical measurements, and we understand the energetics with great precision, and we understand all these different aspects of flipping from B to Z. What about the biology? And so the biology of the zDNA actually has been unlocked by the identification of the zDNA specific domain, the Z alpha domain. And this Z alpha domain is structure specific. It doesn't recognize any sequence specific features of zDNA. It just binds to the backbone. And you can see the zigzag nature of the backbone that relates to this dinucleotide repeat that I talked about before, because these sequential bases have a slightly different conformation around their sugar. And that different conformation causes the backbone to be zigzag, and that's why it's called zDNA. And so Z alpha binds to zDNA in a very highly specific manner. And it was possible to unlock the biology of zDNA by using the Z alpha variants. And so there are a couple of residues that make very specific contacts with zDNA. And by binding variants in these two bases here that exist naturally in humans and looking at what kind of phenotypes they produced when people had those two variants in Z alpha domain, it was possible to understand what Z alpha is actually doing in the cell. And from that, then it's possible to deduce that zDNA is actually the functional element that Z alpha is responding to and is actually controlling the phenotypes that were observed. And these phenotypes were to do with regulation of interferon production. And so if you have the variants, you tend to make a higher level of interferon than if you don't have the variants. So in that sense, the Z alpha domain was turning off the interferon responses, a negative regulator of interferon responses in normal people, but not in people with the variants. So the Z alpha domain was isolated in this enzyme that Sasha introduced, this enzyme called ADA, which is a double-stranded RNA deaminase, and it has a Z alpha domain which binds zDNA and zRNA. And it also has double-stranded RNA binding domains, which are structure-specific and not sequence-specific. And it has this attached to a catalytic domain that performs the editing function. So it's an example of where you're mapping shape functions to string functions. So you have the Z alpha that binds to zDNA, which localizes the enzyme. You have the double-stranded RNA binding domains that bind to fallback structures in RNA that form the double-stranded RNA substrate. And then you have the string operation where you have the editing of adenosine to inosine to produce change in the base at that location. So it's a nice example of where you can use shape and sequence, and you can use shape functions and string functions to produce an outcome. But how does ADA prevent the immune response from happening? So it turns out that there are host sequences in double-stranded RNA that have a propensity to form zRNA. And these sequences are present in host RNAs, but not in viral RNAs, for example. And so when you initiate the response to double-stranded RNAs, which is a natural response because most viruses make double-stranded RNAs, and so that's a warning sign that something's wrong in the cell. So if you get double-stranded RNA being produced, you want to know whether it's a host double-stranded RNA or a viral double-stranded RNA. And so as the response is activated, you have these helicases which then combine the double-stranded RNA, and they actually stretch the double-stranded RNA. And if the host sequence is there that forms the zRNA, some sequences will flip to zRNA, identifying it as a host sequence. Then ADA will bind through the alpha domain to those host sequences, and then will perform the string operation to make edited RNA and get rid of this double-stranded RNA that's causing the problems. Of course, because viruses don't have these Z-forming sequences, then they remain as double-stranded RNA and can invoke interferon response. So it's just a nice example of where you can combine shape and string functions together to produce a biological outcome. So let's talk about the flip-on computational gates. What can you do with flip-ons with this binary 0 or 1 state? And so you can use flip-ons for logical operations, and I'll give you some examples of and or not gates that we can make with flip-ons. You can use them as topological switches that allow you to have action at a distance, and you can use them as state switches where you can switch something on or off, or you can toggle it between states. So here's a logic implemented with flip-ons using RNA splicing. So as I mentioned, you can have alternative splicing that happens. So you can have either inclusion of exon 2 or exclusion of exon 2. So you get alternative products produced. So, and you can have double-stranded RNA that brings the different junctions together that enable splicing. And then you can have double-stranded RNA editing happening, which changes the probability of exon 1 joining to exon 2 versus exon 1 joining to exon 3. So here you have the splice is conditional on Z formation and on binding of the P50 Z alpha containing 801. So if you have both these Z formation and you have the presence of this editing enzyme in the cell, then you can edit the double-stranded splicing substrate and change whether exon 1 goes to exon 2 or exon 1 goes to exon 3. So how can that be turned into a not an AND gate? Well, imagine that exon 2 is actually a kill tag. So if this is present in the RNA that is produced, then that RNA gets destroyed. So that automatically gives you a NOT gate. So if you join exon 1 to the kill tag and then to exon 3, that RNA will automatically be trashed. So you have a NOT gate. So any RNA that has this kill tag will get destroyed. On the other hand, if you have exon 1 connected to exon 3 directly, then that acts as an AND gate. So being able to use flip-ons to change the outcome of an RNA splicing process is quite easy to do. And it just illustrates how flip-ons can control this particular inclusion of this tag. So the flip-on, by excluding this kill tag, makes an AND gate. By including this kill tag, makes a NOT gate. So considering that you have potentially millions of these flip-ons throughout the genome, then suddenly you can start thinking of the genome as a binary genome, and you can start thinking of all these potential AND and NOT gates that are through the genome, and you can start thinking of all the combinational diversity you can generate, and all that diversity changes phenotype, so it can be selected. So it's, rather than thinking of an analog sequence-based genome, you can think of a genome full of flip-ons and binary switches that can change outcomes. So another example is the topological switch, and this alludes to the fact that you can have a sequence that's Z-forming, that when it gets flipped from B to Z, it changes the topology of the whole domain, and that change in topology gets propagated along the DNA and allows action at a distance. So flipping this particular sequence here from B to Z may change the conformation of a sequence far, far away and flip that back from Z to B. So this allows you to have a topological switch where you have input at this particular sequence, changing the output of this particular sequence. And this is occurring over large distances of DNA. So it's an interesting feature of chromosomal organization. And so depending on what sequence is Z-forming at each of these two sites, in this case, we have a very good Z-former. By that, we mean it's easy to initiate the flip because of its sequence composition. And we have another Z-former here, which doesn't actually flip very well, but once it starts flipping, it's long. And so it actually can capture all the topological energy. And the consequence is that when you start talking up this particular closed circle double-stranded DNA, you initially induce the flip in the Z1, C8 sequence. But as you keep cranking it up, suddenly you flip the CA sequence into Z, and this absorbs all the topological energy and flips the Z1 sequence back to B while capturing Z formation in the Z2 sequence. So it's not only a theoretical possibility, but you can actually look at this in gels and actually demonstrate that these two different conformations, these two different Z-forming sequences compete with each other to form zDNA, and that you can switch zDNA from one to the other just by changing the amount of superhelical density in this particular closed circular plasmid. So that's an example of a topological switch. You can also use wrapping of DNA into these nucleosomes that were mentioned before. So normally, because the genome is so large and because a lot of it is non-coding and of dubious value, most of the DNA is actually packed into these nucleosomes. And so this represents an off state. So they're tightly packed into a dense bundle. And this is called heterochromatin. So this heterochromatin exists, but it's not transcriptionally active. It's not contributing to the codon wear in the cell. So it's definitely in an off state. So the wrapping in histones progresses any transcription. So you can use this actually as another source of Z formation, as we mentioned. So if you eject this particular nucleosome, then you flip potentially that sequence into Z-DNA. And it just so happens that, again, we come back with this magical number of 1.6 turns around the nucleosome is actually sufficient to flip one B-DNA helix to a Z-DNA helix. So this is another way of using histones to regulate gene expression and also to regulate flip-on conformation. So if you start thinking of nucleosomes like in this context, you think that the nucleus has a whole bunch of energy stored in these conformations and these structures that is waiting to be unlocked. And if you can unlock that energy, then you can stabilize these alternative DNA conformations and you can use it to do interesting biology. So this can be used, for example, to eject the nucleosome, and this generates energy to generate open chromatin, and then this can be used to actually then this energy that's stored here then can be used to assemble the different transcriptional factors that are necessary to guide readout of that particular chromatin region. And so here, Z-DNA is just acting as an energy store. So it captures the energy from unwrapping the nucleosome, and then it uses that energy to help power the assembly of these enhanceosomes. And even though we're just talking about one factor here, these enhanceosomes themselves have structure and they involve a lot of negative supercoiling of the DNA and wrapping it around different components. So this, in this case, the flip-on acts as a store of energy to enable this transition from one state to another state. And also you can change out one particular enhanceosome binding factor to another just by using the same mechanism. So you can kick this off, capture the energy as Z-DNA, and then use that energy to stabilize a new conformation there. So just to compare the different types of logical operations, shape versus sequence, you can see that sequence has high specificity. Finding it is search dependent. The outcomes change with sequence variation, and there's competition between the different target sequences that you're searching out. In terms of structure, it's much less complex. You get binary outcomes, and you get confirmation between competition between conformations. So there's lots of variations on the themes there. You have other flip-on types. You have the string structure functions that I talked about, and then the search algorithms that are involved that underlie finding information in the genome. So the G quartets are these four-shaded structures. They form the sites of open chromatin, and they're associated with gene regulation and DNA repair, and they're very stable once folded. Triplexes allow you to put a third strand into bDNA, and that helps localize whatever's attached to that third strand. And so that third strand can have lots of different potential motifs that can be recognized by proteins, and so that you can get a lot of combinatorial diversity in the proteins that you actually attract to that site of triplex formation. So that's another example of how you can use an alternative conformation to localize cellular machinery to a particular genomic site. You can use strings to create structures, and so you can have complementary strands that base pair with each other, and they combine to form a structure. And why is that useful? Because it allows you to use structure-specific proteins to localize that have particular patches on them that allow you to localize transcriptional machinery to that particular structure. So the genetic code is a fine example of this, where you have core codon machinery that reads out the RNA from the genome, and you have core translational machinery that takes the RNA and converts it to protein. But it all depends on actually making a shape between the nucleotide triplet and a tRNA for this particular core translational machinery to engage. So using generic machinery is possible just because you actually convert the sequence into a shape that's recognized by the generic machinery. And this is not only true of the genetic code, but it's true of other regulatory RNAs where the adapters can be microRNAs that control RNA stability, or they can be guide RNAs that control CRISPR-based immunity. So here's an example of just going from string to structure to function. So the last thing I just want to cover is the type of strings that are called repeats. They're very frequent in the genome. They are low complexity. They repeat the same sequence, like poly A, poly G, alternating CG, etc. They don't have much information in them because they have low complexity, but they're actually very useful for facilitating search of information in the genome. And so these low complexity sequence repeats can make structures, as we've been talking about, that make flip-ons. They can actually tag RNAs so that you can tag a bunch of unrelated RNAs with the same particular low complexity sequence so that they can be co-regulated. And they also encode peptide patches on proteins. And that can also help these proteins find other proteins that may be necessary to help assemble a biological machine. So each of these different types of outcomes from simple sequence repeats allow search. So you can search by structure, you can search by target, and you can search using these patches, these peptide patches on proteins. And so the indexing varies by where these sequence repeats act, whether they act on DNA, RNA, or protein. And the location within DNA, RNA, or protein actually determines whether they are active or not. And you then have natural selection to maximize the efficiency of the search function in terms of trying to find a particular location or a particular target, or to build a particular biological machine. So we haven't mentioned peptide patches, but they're kind of really interesting. Peptide patches made by low complexity sequences enable the assembly of lots of different biological machines, and this is the way they work. These low complexity structures actually don't actually fold into anything. They can have lots of variability in their conformation, and they can wave around searching for things in their immediate environment. You know, it's just like putting a huge set of tentacles out to find something that's of interest. And when they find something of interest that's complementary, they can fold, as seen here for this protein, and into a biological machine that performs a function. So these peptide patches, these low complexity sequences that don't fold, actually enable search between different proteins to enable whatever function that this protein has to be brought to in close proximity to whatever function this protein has without actually affecting the protein function at all. So these are just tagged onto the core functional domain, and they're just low complexity regions that help these different functional domains find each other to make a machine. So that's it. So I convinced you that flip-ons are a great way of encoding genetic information. They change how codons are used in the cell. They change how the wetware is assembled in a cell, and because they change phenotype and change biological function, they're subject to natural selection. So then actually elements of genetic elements of a multicellular genome. So thank you very much for your attention. I'm sorry, I think I've been a little bit over time. Oh, no, 5 minutes.
No problem, no problem.
Yeah. So happy to take any questions if, as you have them.
Yeah, thank you so much, Alan, for a comprehensive presentation. It's, I hope everybody get a lot of new concept. Are there any questions we can catch from the audience? Actually, I have, you know what, I read your concept about paper, stone, and seeders some times ago in your immunity paper, and I just recall, maybe we never discussed it recently. And my questions, can you give an example? Did you call it introgressive logic? Am I right? Or
intransitive logic, yeah.
Intransitive,
Yeah.
Intransitive logic. Can you just give a biological example? So it's an interesting concept when one object can be both winner and loser, right? Depending on
yeah, I can. So if you think back to that the diagram with the 4 cells in it.
Uh-huh.
And so that's purely intransitive, because if one cell kills the next cell, then that cell is no longer present to kill the other cell. And so the cells at the corner end up dominating, and the cell at the lower corner can kill the... So again, depending on which cell dominates in that thing determines which the response is. So depending on the context, you can change which cell dominates. And what's kind of interesting is that you can switch from one stable state to the other stable state, but the transition in between is really chaotic. And there's actually a figure in the paper that actually shows you how the transition occurs in a crazy kind of manner. And at one point it's highly unstable, and at that point, that's where the switching occurs, where you can go from one to the other, and that's where the context has its effect. It just changes the outcome of that chaotic process.
Yeah, it's a cellular type level. Do you expect anything like that at the genome level?
Yeah. So
typically with sleep-ons.
No, actually, I have an example too, which I'm working on at present. That you can make it work in the cellular level too, and a genomic level as well.
Genomic level, so it can be found in at the genome level too.
Yeah. So anything that's context specific, anything that stabilizes the a flip on one conformation to the other, can initiate a process that inhibits the B-DNA dependent outcome. And so, and likewise, the B-dependent outcome can inhibit the formation of an alternative conformation. So you can change state that way, and it's also a chaotic transition as well.
Yeah, it's interesting that actually once you have this element described and formalized, then the next level you can build any circuits of your wish biology circuits, as you started to present. And what we expect is probably there are much more than the just binary circuits like AND and OR, since the biology has more states available rather than 2. And that would be an interesting subject.
Yeah, and what's interesting too, if you want to build on that concept, because of all the potential for the binary switches that are in the genome, there's probably never any 2 cells that are in the same state. And so what you end up doing is you end up stabilizing one rock, paper, scissors state or another in each cell. And then within a tissue, you can select those cells for which the response is most appropriate. And so you're going straight from the switching within the cell that establishes potentially an infinite number of different states within a cell, and select those states that are most adaptive to the particular context. And when you're done with them, you just destroy them and start off again with some fresh cells. And there's lots of
and what you have recently discovered that this prisoner dilemma is also applied to human living. So not human living, but cell options to go. So there are many logical experiments that probably we can conduct to explain the behavior of inner cell at the genomic level, right?
Yeah.
So all these logical experiments are kind of scalable at different levels. So level of humans, they're applicable to level of a cell or level of a genome to determine the behavior.
Great things about biology is because the system is self-replicating. What you can find those things by designing genetic screens to show those effects. So, unlike a computer program where you keep plugging away until you get the outcome that your supervisor wants, here you can just set the system going and bias the transition probabilities at the start. So you end up selecting those that are optimal for the outcome that you've specified. Mm-hmm. So it's all self-contained. And so even though you never get the same answer twice, you end up getting some really, really interesting answers and really interesting solutions to problems that you would not otherwise have come up with just using transitive logic.
Mm-hmm. Cool.
So yeah. That's the joy of biology, is it's full of surprises.
Yeah, that's true. Any questions from Sasha, maybe Dmitry?
Well, in fact, in optimization, we have a heuristic called evolutional optimization, where the problem is encoded as a set of binary switches, and these switches can be evaluated for a target problem, like what combination of switches is the most suitable for a given problem. And well, the main issue in this optimization is to actually encode your problem in switches, then let it evolve like in real population, but by modeling in a CPU on a computer, like how these switches evolve by mutations, crossing over, etc., and then estimate the fitness of each population in each turn, and select only those which are the most fittest, then repeat. And actually, if we have an option to build something similar inside a cell, inside a living cell, and I believe the main problem here is to actually somehow measure this fitness of a particular combination of these binary switches, like Z-DNA structures maybe, and well, map these binary switches to a fitness, and fitness should be relevant to our problem. If this problem is solved, then any optimizational problem which can be reformulated in these particular terms will be solved a lot more efficient than it is solved today, because living cells can divide rapidly, and it's like a parallelization 100 and 1,000 times more than in a supercomputer, because you can have a bunch of pallets and a bunch more and a bunch more. It's not a big deal. So yeah, once we have this way to formulate the target problem as a fitness problem for cells, then we are in a really good shape for applying it to real optimizational programs.
Yeah. Sort of abstracting a little more than that, you definitely have a lot of different implementations of Markov models which look at transition probabilities. And I think the advantage of the biological system, as you indicate, is that it's possible to select a Markov solution that is initially very rare, and because so it's like a needle in a haystack, and it's possible to select out that solution from all the other competing solutions. But it's easy to do in the biological system, but hard to do computationally.
No.
It's time consuming, right? So parallelization is enormous in living systems. Cells like 1,000, millions, it's not a big deal, right? You can grow it. That's it.
Yeah, and plus you can keep introducing variations in that process as well. So you're not stuck with the initial starting conditions. You can continue introducing variations and changing transition probabilities during the selection process. So, and each time you may introduce another 10% of variations. And once you start going through 6, 7, 8, 50, 1,000, 1,000,000 steps, the combinatorial diversity becomes huge. And, it's a computational problem that's insolvable, but in terms of a biological solution, a subset of those spaces can be easily explored rapidly, because you're not starting off with all the potential combinations at once. You're expanding them as it goes, and the combinations that you're introducing, or the variations that you're introducing, are dependent on the ones that you had previously. So you're compounding the variation rather than using it as an initial state.
Yeah. The only problem left is to actually encode some optimization problems, other fitness measure or something like that.
And there are many problems with experiments with genetics. Computers are only in fetal stage, embryonic stage.
Yeah, there was that attempt back in the 90s to solve the traveling salesman problem by using DNA encoded.
It took him for a week, I guess, Adelman's problem.
And it involved large test tubes to contain all the different potential variabilities that he could have. But again, he was starting with the whole space defined ahead of time, and with all the transitions specified, from A to B to Z to D, and biological systems don't know what the potential cities are to visit. They find them as they evolve. They don't start with a defined goal of making an arm or making an eye or something like that. They just discover that outcome as a result of their search.
That's true. Yeah, with now at our stage of development, the solution is found immediately. The only problem for us is to extract. Experimental technique is not that advanced that we can extract at the same time as the cell finds solution to our problem, even if we encode it. Yeah, and also I see the development. So now what we are doing, including our lab, is to mapping all these, flip-ons, all these DNA structures, and trying to find all the relationship with epigenetics code, how it works. And maybe the next stage, once we've done it, we can find these, these circuits, logic units of cell operating by example, and maybe we can extract logic we never thought of. So we know binary, we know this concept of see this paper stone. Maybe we find something we never, we just don't find.
Yeah, so the binary logic won't change, but the transition probabilities will change. So I think we'll probably be doing a lot more Markov modeling to try and capture that information.
Yeah. So life is interesting, science is interesting, and never ends up. Okay. So unfortunately, we cannot take, this is actually the bad side of the concept of streaming. At least we cannot take questions, but please do not hesitate to email us and or come by and just ask question in person or email or any other means of communication. And I hope everybody enjoyed the presentation. Thank you, Alan, for this keynote and very comprehensive talk. At least it's a real generalization of, and a high level generalization of what we are doing and trying to answer side small questions. Now you presented to us the entire picture and the goal, the ultimate goal of what we have to decipher.
Thank you very much for the opportunity to present. I really enjoyed putting the presentation together, and kind of learned a lot myself in trying to understand the questions that were important.
And by that, I end up the session, and have a nice day.
Thank you very much.